it is putting on impactful strides in integrated genomics and computational biology. Over to you, Dr. Sona, and we will complete that talk within 15 minutes, and we will take 5 minutes of questioning for both these papers.
So good afternoon, everyone. I think everybody is eagerly waiting for their lunch, so I'll just run fast to my presentation. So I am working here in Indian Institute of Spices Research, Calicut. I think many of you might not be aware about this institute. So those who are visiting Kerala, they might have definitely gone to Wayanad. So on the way to Wayanad, there is one institute on the right side of the road. This beautiful place. Anybody who is coming over can definitely visit the institute, and we'll be happy to show you around. And the most interesting fact which I feel about my institute is like we have a bioinformatics facility. So it is for the bioinformatics and integrated facility where we host, where we do several things like we conduct trainings, we mentor students like MSc and post MSc students, the students who actually who have done bioinformatics or some other area like molecular biology, microbiology, and who need exact training in bioinformatics. We are supporting so many students as well as faculty here. And another important part of this facility is like anybody who is interested in bioinformatics, they can attend online trainings as well. We give hands-on support to the students who are interested in online trainings, and those who are not able to come and travel to this place, they can always avail this facility, which is the trainings are conducted in 6 months gap. So anybody who is interested can come and join us. And when I see the students, many of the MSc or the PhD students, they are having lot of data with them, but they are not able to present it in a way our speakers have presented in the previous sessions. So anybody who is interested in science, they must be able to visualize, they must be able to represent their data in a very perfect manner. So after conducting each and every trainings, I used to get calls from different people like asking me, Madam, I have this data, I need to represent it, I need to show it in the best way as possible. But as the number of calls or number of such requests came, I thought like why not create a software or any tool that can actually help the experimenting scientists, because they have to spend a lot of time trying to implement a tool, like even if it is an algorithm that is present in GitHub, it would be difficult for them to download and understand how it works, and how difficult it is, like maybe for a non computational biologist, it's difficult for them to download the tool and try to implement, coming up with so many errors on the way. So I thought like why not implement something for the people. First I tried to implement this for the scientists who are working here in the institute itself. And so as we all know that you are aware about the sequencing technologies that are available today, you will be flooded with a lot of data. The only thing is how to represent it, how to visualize it, how to analyze it. These are some of the questions that we usually face. And as far as my institute is concerned, like I'm getting so much data from genome sequencing, then from the transcriptomic studies that are being done on the spice crops, then also on the soil metagenomic studies. Actually, I'm flooded with a lot of data. So as the size of the data increases, it becomes more and more difficult to analyze it. So I thought, why not create something that is useful for everybody, and everybody can use it and do their stuff. So these are some of the common transcriptomics and metagenomic workflows which you will be encountering like when you are doing data analysis. So the initial steps from the cleaning of the reads to the assembly, those are done on Linux. But when it comes to data visualization, we have a strong and powerful system which is called, or statistical package which is called R. And I think many of you will be aware of it, and most of you might have used it. But this is for easy use of R for the PhD students and MSc students who are just trying to improve their programming skills. Now, after completion of any of this analysis, we'll be getting what is called, what will be there in an Excel sheet which is called the count data, or if you are doing some metagenomic experiment, you will be getting what is called the OTU table. So this is where the game starts. So these are the things, like these are actually files which consist of almost 30 to 50,000 number of rows. And then and as the number of rows increases, the ability to identify what you want to show, exactly you want to pinpoint in that data, it actually diminishes. So that is where we can help. So I have designed a small data analysis and visualization toolkit that can actually take in public data or as well as your in-house generated data, which is actually the count data or the OQ table, and it can analyze several aspects of this. Like I have right now designed this tool to include some of the tools for basic statistics, because that is the first thing that we usually want to do about the basic after receiving the count data. Like you will be trying to understand in what range the values will be spread. So it gives a basic statistical idea of your data. Then tools for normalization. Then for data visualization, usually you will be definitely you will be visualizing some part of the data, and you will want to show it as a high quality image in your publications. And for differential expression and for giving the correlation profiles. So this is the current stage of the toolkit. So I'll explain a few modules, which I say that these some of these modules which I have developed, so that you can also just try to, if you are good in programming, you can also just try to create some tool like this. So this is the toolkit. Actually, we have created this toolkit using the R-Shiny package, which is a very powerful and efficient package. If you are able to use it, you can create wonders using this package. And this toolkit actually depends on 43 libraries, and it has a user interface as well as the server side for this app. And you can have conditional input modules in there. So this is the basic interface. You have an option to select which module you want to use, and you can upload your expression file and select the options available. Now this is one part of the visualization module. When it comes to visualization module, as Dr. Sajita has previously pointed out, we have high multidimensional data. So when it comes to multidimensional data, we have to reduce the dimensionality by using several techniques. PCA is one common technique, and we also have techniques like TSNE. So I have implemented both in this Shiny package. So this is the interface. Once you upload the data, it will get displayed here. It will get displayed. And so this is the way in which you have to give your input file. So just an Excel sheet, or you can just create it as a CSV file or a text file and give it as an input. So your output will automatically be displayed here. You can download it, save it, and use it. And this is for correlation, for calculating the correlation when you have a gene expression data. Sometimes you might be wanting to find the correlation between the genes, or maybe you have to find the correlation between the samples. So whatever you want to find, you just have to create the file accordingly. For correlation analysis, you need to have one file like this. So this is for metagenomic data, just an example. So in case of metagenomic data, you need to prepare two files, one like this and another one consisting of only the first column. So you can give it as an input, and you can select what is the type of correlation you want to calculate. So this is the basic interface on how to do it. So you will get the results of the correlation as earlier I have shown a file that can be downloaded. Now coming to this one, most of you might have done differential expression analysis using DEC. So I tried to implement this file, this DEC package in R-Shiny, and this is how it is done. Like you can select differential expression module and upload the count data or any the file that you have in the text or the CSV format, and then select the control samples and the treated samples, and you can set the cutoff value. So these are the basic things that you use when you are doing DEC. And you can visualize your result in the form of a heat map or a volcano plot, whichever is needed. So this is a small part of the application that I have been developing, and this is very useful for PhD students. So once we publish it, we'll be definitely giving out the link to through different groups. And so as a conclusion, like most of our efforts as a computational biologist, the most of the efforts focuses on how to simplify very complex gene expression data, and to enhance the data interpretation, and for quality analysis, and for the integration of multi-omics, as we have seen in the previous sessions. And most importantly, whatever tool we develop, it should be very much user friendly, and I believe that we can make a huge change in the community by bringing up this tool. Thank you.
Now we open the session for the questions for both the speakers, Dr. Sona and Dr. Renuka.
I have a question. My two questions. This is a web-based downloadable one.
Right now it is not uploaded. Like we'll be uploading it in our GitHub page in the developer mode. But once it is done, once it is something, I would like to add few more modules, like to select the colors, show me other ggplot options to this, and then I will be uploading it.
How much the input file I can
So it's very simple. I have shown, no, like in that one count file, which is should be in the TXT format or in the CSV file format. You can just simply upload it. Upload options are there. Yeah.
One question to Dr. Renu, one question to Dr. Sona. Dr. Renu, when you did the linked RNA protein interaction, there's a Zucker-Turner model for RNA structure. So it's a Zucker-Turner model.
Please keep it the mic close to your mouth so that we can all hear your question.
So there's this Zucker-Turner model for RNA structure. So the Zucker-Turner model can be extrapolated to any type of RNA like linked RNA or myRNA or all types of RNA.
No, we have taken the whole ANC RNA which we have the sequence and then tried to look into the structural aspects of.
Within the structural aspect, the Zucker-Turner model is a template model for RNA. So can it be extrapolated to any kind of RNA like myRNA, Sona RNA, 5 RNA? Dr. Sona, the question is to use the R-Shiny, can it also install bioconductor package?
Extra?
Bioconductor package.
Yes, it has so many other bioconductor packages within the library itself. So I have included that also. Because without any other package, we cannot implement such a tool because we are taking it from further volcano plot, for example, we are taking it from the enhanced volcano. But we have not implemented further beautification options, which will be coming up in the next version, higher version.
And just like women in bioprocess there, just for community, this is rladies.org.
Yes, I know. Yeah, thank you. So I would like to thank the organizers for inviting me to the session. Thank you.
Since that is Y chromosome is one of the reason of disappearance, Y chromosome disappearance is one of the reasons of prostate cancer or not, or and how.
Can I answer that question? Or you want to answer?
You please.
See, the disappearance of Y chromosome has nothing to do with cancer prostate. Y chromosome is getting decrease in size over the generations. And we know that after few days, there will be no male species on the earth. Okay. So, yeah. So then there is no question of prostate cancer after that. So we will be summarizing each one's talk. I will be doing the first three and Madam will be doing the next one. So Dr. Sarjita Lulu, she has been talking to us about tumor microenvironment through single cell RNA and spatial transcription database. Tumor microenvironment, molecular immunology, cellular components of TME, non-cellular components of TME, and cytokines which are from the TME, that is both types, promoters as well as inhibitors. And she spoke to us about those personalization, which actually I have been thinking of over a long time. That not every two persons having the same kind of cancer require the same dose of medicine or radiation. So that is there, and then she has told us about molecular and cellular profiling and special transcriptionase. Then she told us about data processing, quality control. Quality control is there every part of this process. Horizontal and vertical integration of those two things. Then cell-to-cell interactions. Actually, tumors are nothing but a loss of cell-to-cell interaction. So that is also dealt with. Then she has told us about the spatial heterogeneity of tumors. Then the second speaker, Dr. Rakesh Kama, whom I'm acquainted with and I've seen lot of times. He spoke to us about human migration and biodiversity and genetic affinities and genetic variants of the Himalayan and sub-Himalayan populations, that means the adjoining population. And he said that he took samples from 3 generations of people like Chompa, Bhutia, Sherpa, Limbu, Leka, Tamang, Nishi, Mizo, Naga. And I lived with each one of them. So I know what he is talking about. Okay, and it's not easy to differentiate for other people. Mainland people like my husband cannot differentiate between these people. I can differentiate between each one. So I see, I said, this is Naga. That one, okay, so that one, that's Lepcha. So my husband asked, how do you know? But I can. So they are all actually different populations, and he has called about the 3 markers, that is Y chromosome, mitochondrial DNA, and high density autosomal SNPs. Since I have done this kind of job, I know what he was into. It's massive amount of that, and in the pre-AI eras, it was very difficult to do this. Then he has told us about the East Asian groups and how the gene flow was there. And then he has told us about exons 3 and 4, the mutations, and how there was low activity of those. And then about the lactase gene. That was something which I didn't expect him to do. But lactase gene, 14 kb upstream, he has spoken about it, and he has told us about the migration which he thinks is from the east towards this area, that is from Japan and Thailand and all those areas towards mainland. This sounds very funny, but in our churches in Kerala, we have kept old records saying that people have migrated from there. This is written records on farm. And I was not believing it, and now Rakesh has given me a clue that this may be true. Okay, thank you. Again, over to Dr. Charu.
Followed by this, there was a talk by Dr. Ranjit Das, where he has shown the, basically the connections between pneumonia, heart failure, Parkinson's disease, chronic fatigue, and COVID-19, and using participants, genotyped them, and showed all the details. And this talk was followed by Dr. Nedka's talk, where she has identified the potential biomarkers and phytochemicals for prostate cancer. Hello. Yeah. After this, Dr. Renuka has identified potential biomarkers and phytochemicals for prostate cancer. She has given all the details of the prostate cancer and how she has used the in silico approaches for the identification of these biomarkers. And this Dr. Sona, definitely she has done a lot of good work like other speakers, and she has developed a toolkit for interactive downstream analysis and visualization of NGS data like transcriptomics and metagenomics. For this, she used our Shiny package, and I think with this, thank you for all of you to be patient.
I think with this, we have come to the end of the session. But before we conclude, we would like to express our heartfelt gratitude to our speakers who have enriched us with their exceptional research and perspective. May I request our session chairs to kindly present the token of appreciation to our speakers. And for that, I would like to invite all the speakers of this session to kindly come upon the stage. Professor Sanjita Lulu for her insightful talk on decoding the tumor microenvironment using advanced transcriptomic techniques. Dr. Renuka for her innovative approaches to identify biomarkers and phytochemicals for prostate cancer. And our last speaker, Dr. Sona Charles for her presentation on R-Shiny-based toolkits for NGS data analysis and visualization. And Dr. Rakesh Samal for his fascinating genomic exploration of the Indian Himalayan population. We would now like to honor our session chair, and may I request Dr. Marina Banerjee to kindly come upon the stage. Once again, a big thank you to all our speakers and chairs for their valuable time, knowledge, and appreciation. Let's give them a big round of applause. With this, we can now move for lunch, but I would like to announce that please be back at this hall at 3 p.m. sharp. We will strictly start the session at